The thought of DNA began when Charles Darwin published his book, The Origin of Species. The book was a compilation of his studying over the years and proposes Darwin's theory of evolution by natural selection, descent with modification. Many people were skeptical of his ideas because he challenged the widely accepted concept that God created everything in 7 days. An Augustinian monk known by the name of Gregor Mendel conducted many experiments on pea plants and gave rise to genes, dominant and recessive traits, and heredity. In 1866, he published a paper that described his theory of invisible factors that are responsible for the visible characteristics that we exhibit. Although today he is known as the father of genetics, it took almost 3 decades for his paper to finally be taken seriously. In 1869, Friedrich Miescher, a Swiss physiological chemist, identified what he called nuclein in the nuclei of human white blood cells. While he was attempting to do more research on his findings, he realized that he'd found a completely new substance, but the scientific community didn't widely appreciate his work for more than 50 years. The eugenics movement really defines the lack of understanding that the world had of heredity and breeding. Mendel's theories were rediscovered. Researchers became interested in selective breeding of humans once they learned that they can use math to predict offspring genetics. The popularity of eugenics began to decline around 1913. By 1944, it was common knowledge that genes were the discrete units of heredity. Finally, a man named Oswald Avery identified DNA as the transforming principle. Avery was an immunochemist at the hospital of the Rockefeller Institute for Medical Research. He did lots of research with bacteria and discovered that bacteria were not proteins or carbohydrates, but actually nucleic acid, which he then discovered was DNA. Avery and his colleagues published a paper in the Journal of Experimental Medicine in 1944 that explained how DNA was the transforming principle. The paper inspired more research, although it was not widely read by geneticists. After scientist Erwin Chargaff read Avery's paper on DNA, he was inspired to completely turn his research to study the chemistry and composition of nucleic acids. The first thing he did was try to find a way to analyze the nitrogenous components and sugars in DNA from different species. In 1950, he had 2 major findings. 1. Any double strand of DNA has an equal number of guanine and cytosine units and an equal number of adenine and thymine units, and 2. DNA composition varies among different species. Rosalind Franklin worked with Maurice Wilkins, a scientist, and Raymond Gosling, a student, to produce 2 sets of high-resolution photos of DNA fibers. Franklin used the photographs to calculate the dimensions of the DNA fiber and to determine that the phosphates were on the outside of what was probably a helix structure. Despite her findings, Thomas Watson and Francis Crick published their same discoveries before she did. In 1951, Francis Crick, a professor at Cambridge University, met James Watson at the university, and the two began to study the structure of DNA. They used X-ray data and model building to discover the double helix structure of DNA. They wrote a paper on their findings, which was published in 1953. In 1962, they and Maurice Wilkins were awarded the Nobel Prize for Physiology or Medicine. In 1959, geneticists used cytogenetics to discover the link between chromosome 21 and Down syndrome. Stains were developed so that scientists could bind the stain to the chromosomes and be able to identify which chromosome was where and if there were any abnormalities. In 1957, Marshall Nierenberg, a scientist at the National Institutes of Health, dedicated all his studies to nucleic acids and protein synthesis. In 1961, Nierenberg and his colleague conducted an experiment that showed that a chain of repeating bases uracil forced a protein chain of a repeating amino acid. They conducted a few more experiments before they published their papers, which showed the proof that the genetic code could be broken. In 1965, Nierenberg became the first person to sequence the code and was awarded the Nobel Prize jointly with Robert W. Hawley and Har Gobind Khorana. By the 1970s, molecular biologists were able to decipher the genetic code and read the amino acid sequence in proteins, but still not in DNA. Cambridge graduate Frederick Sanger could identify the free amino groups in insulin, and he concluded that if proteins were ordered molecules, then so was DNA. He worked with RNA sequencing because it was smaller, and the same technique he used is now used for sequencing DNA. In 1988, the National Research Council created a program to map the human genome, and the Human Genome Project began in 1990. The U.S. Department of Energy and the National Institutes of Health published a 15-year plan for the project. The project's goals were to identify all 3.2 billion letters in the human genome, mapping and sequencing other organisms' genomes, and developing technology for the purpose of DNA mapping. Dolly the sheep, the first mammal ever to be cloned, was cloned in 1996 from an adult cell. Dolly was the only lamb to survive out of 277 attempts. While other mammals had been cloned from embryo cells, Dolly showed that cloning was possible with an adult cell. In 1999, the first human chromosome was decoded. It was chromosome 22, which contained 33.5 million chemical components, or letters. In 2000, scientists finally decoded the genetic code of a fruit fly. This is important because fruit flies have very similar biology and development as humans. In 2002, a mouse became the first mammal to have its genetic code decoded. It was discovered that 90% of a mouse's genome can be compared to a human's. In 2003, the Human Genome Project was completed. Scientists discovered that the final form of the genetic code has 2.85 billion nucleotides, and the predicted error rate was 1 in 100,000 bases sequenced. DNA Worldwide and Eurofins Forensics were the first in the world to prove that identical twins didn't have 100% of the exact same DNA in 2013.